Okay, you can see it now.
Thank you for the invitation. I'm very honored to participate in your conference, and we start to discuss about the prevalence of infertility. It is an increasing disease in all over the world, and in Europe. You can see in this map the different incidence of the fertility rate in the different countries of Europe. And the cause of infertility, we know all the causes of infertility. We have 30% of male infertility and 15% of female infertility. But we have a very high percent, 25% of idiopathic infertility. This is a very high range of infertility that we can do anything. We can only try to add something, to try something of ART to obtain a result. What we can do for treat the infertility? We can use medical treatment for ovulation disorders. We can try with surgery, tubal, very rare, or uterine surgery for polyps or myomas, laparoscopic evaluation and treatment for endometriosis, or the thing that we can do very often, ART, assisted reproductive techniques, that consist in a lot of different techniques that can help us to obtain a pregnancy. We can use both in hypothalamic pituitary insufficiency, gonadotrophins, and also in hypothalamic pituitary ovarian dysfunction, like PCOS. After we try with clomiphene citrate oral drug, we can, when we have no results with clomiphene citrate, we can induce ovulation with gonadotrophins. In ART, gonadotrophins are used in all the different methods. The methods most frequently used are IVF, in which gonadotrophins are used before oocyte pickup to facilitate the development of a lot of follicles, controlled ovarian hyperstimulation, and in very small cases, when we use this, in intrauterine insemination. We know that gonadotrophins start in 1960 with the first human menopausal compound. After 28 years, we had the first IVF baby birth. Then we purify the urinary gonadotrophin, the FSH, and then in 1992, we had the first recombinant human FSH. 1997, we have the first baby with the use of recombinant FSH. After this, in 2014, ended the exclusive patent of recombinant FSH, and there was the approval of biosimilar FSH. There is, we know that there is a difference between recombinant versus urinary FSH. There is a lot of papers on this. We know that recombinant FSH require low doses and short duration to obtain a good result. And urinary FSH has a variability and probably something that human that can stay in the different product. What are a biological drug? A biological drug is a molecule of very great size that results from living cells and can use it for treatment, diagnosis, of prevention of some disease. There is a lot of different drugs. We know in this period DNA vaccines, and we know that there are a lot of conditions that can differences the manufacturing and the production of these biological drugs. And these are very more difficult than that of traditional drugs. And also the response of the patients depend of the production and the different way of production of biological drugs. We know that the biological drugs are produced from cell substrates by genetic engineering techniques that use recombinant DNA technology that requires cloning to join DNA for various forms and spaces to obtain a cell line expressing a specific protein that can be used for treatment. In our case, the recombinant follicle stimulation hormone is similar to the natural FSH that is expressed in humans, but was generated by Chinese hamster ovary cells. For the production of biological drugs, we have a lot of different steps of the process that every steps of these can influence the characteristic of the final product. And we know that the World Health Organization defines biosimilar, a biotherapeutic product that is similar in terms of quality, safety, and efficacy of the biotherapeutic product already authorized. There is a very great difference between the generic drug and biosimilar drug. Generic drug has the same as a simple chemical structure and is identical to the reference product. Biosimilar is a biological drug and are not only similar, not identical to the reference drug. And for this reason, every biosimilar have a different degree, a small degree of differences in between the reference product and the biosimilar. And every biological product produced by different manufacturers, different from each other and from the original product. For this reason, biosimilar must be evaluated on the basis of analytical data, clinical and non-clinical, to be considered as similar and to an original biological drugs. This one is very important because we know that the biosimilar has the same efficacy or ways of very similar efficacy, but is demonstrated that there are differences between the biosimilar and the originator. It's very important that the differences do not be clinically significant. The European Medicines Agency, in evaluation of biosimilar, requires that there is no significant differences between the originator and the reference, the originator and the biosimilar in terms of quality, effectiveness, and security. And the comparison with the reference product is, includes studies concerning quality to confirm that the biosimilar have the same chemical structures, studies on safety, on efficacy, and on to verify the potential immunogenic risk of the biosimilar. The biological activity of the biosimilar is due to the production, the purification, formulation, handling, and storage, and this influence also the effectiveness and the safety of the product. We have to understand very well pharmacokinetic and pharmacodynamic the biosimilar drug, and is necessary to have a pharmacovigilance plans for the 2 years after the drug has been marketed to know if there is differences and side effects that are that could be very important. The first FSH in the market was Ovalip in 2013, and after this starts phase 1 study that want to evaluate safety, tolerability, and pharmacokinetic of single ascending dose of this drug, starting from 37.5 international unit in ascending doses can produce some negative side effects. We are using 40 healthy women from 18 to 39 years old in the screening, and start after a downregulation at day 21. We see that the dose of recombinant biosimilar recombinant FSH are very clearly demonstrating the blood sample of the women without any important side effect. There is the only little side effect that during treatment there was headache and dizziness and no safety aspects or outcome related to the dose from the vital signs of the patients, ACG and laboratory parameters, and objective examination. For this reason, safety and tolerability of the biosimilar drugs is the same of the originator. And then was evaluated the bioequivalence of the biosimilar on the originator in 36 healthy women. And we can see here that the equivalence, the bioequivalence is very the same between the biosimilar and the originator product. Then we have phase 3 study to determine the safety and the clinical bioequivalence of the biosimilar than the originator in infertile women with ovulatory cycle to go to PMA cycle. That this was a multinational, multi-center, randomized, controlled, blinded, parallel group phase 3 study in which was examined 299 infertile women with a body mass index between 18 and 29, 18 and 37 years old, and with exclusion criteria only it was 2 previous consecutive cycles of in vitro fertilization without more than 3 miscarriages, ovarian cyst, estradiol more than 50, clearly pregnancy test positive. Primary endpoint were the number of oocytes. This is the very primary endpoint. Secondary endpoint is the dose of recombinant FSH used, the fertilization rate, and the clinical pregnancy rate. The treatment is the classic long protocol with GnRH agonist for the suppression and a fixed dose with 150 unit international of recombinant of originator and biosimilar for 5 days from the first day of the menstrual cycle, and then at day 6 there was a dose adjustment, and then final follicular maturation with ACG and oocyte pickup and evaluation after 19 days from the oocyte pickup of pregnancy, and evaluation clinical pregnancy ongoing. And the baseline demographics characteristics of the patients are the same in the 2 groups, and the same was the cause of infertility in the 2 groups. The results was that the number of sites recruited was similar in biosimilar and in the FSH originator. There was no significant differences between the dose required, no significant difference between the dose range in the 2 treatment arms, and the percentage of patients for whom was necessary a dose adjustment was slightly lower with biosimilar compared to originator. And same, the number of days required for the stimulation are slightly less but not significant for the group treated with the biosimilar than the group treated with the originator. Oocyte quality was the same in the 2 groups, and around the 20% of those sites were is the best stadium, 40% stadium 2, and worse. There was no statistically significant differences between the two groups in terms of number of biochemical pregnancies and clinical pregnancy. And for this reason, we can confirm that there are no differences between the originator and the biosimilar FSH recombinant FSH. Primary and secondary endpoint in the two groups were respected. Similar numbers of sites, and very important, the pharmacodynamic similarity was confirmed, safety and tolerability was the same, and there was no unexpected event in group of biosimilar drug. When we have no pregnancy, we can try for the second or the third cycle, and there was no differences between the originator and the biosimilar in terms of clinical endpoint security and in terms of efficacy. This is a new paper that evaluate in a multi-center randomized phase 3 study the efficacy of biosimilar and the original folitropin alpha. And the conclusion are that the number of sites was significantly more in the biosimilar if compared with the originator. The other secondary endpoints are similar. There is no differences between the biosimilar and the originator in terms of number of follicles of more than 16 millimeters, total dose of SH and days of stimulation, biochemical or clinical pregnancy, and delivery rate. We know that IRT has a cost that is for a great percent due to the drugs. Gonadotropin in all anovulatory infertility treatment is more than 50%, and IVF cycle there is around 40% of cost that was done to the gonadotrophin. For this reason, the use of gonadotrophin that has less cost with respect to the originator is an important is very important in obtain a cycle that is less expensive than another cycle. We know that in Italy the originator after the introduction in the market of the biosimilar has reduced his price around the same price of the biosimilar. And, but in Italy we have a different kind of way. In the public center we have to perform an evaluation, and we have to use the recombinant FSH that is the less expensive in relation of the other. And for this reason, in all the Italy, we can have some region that use one biosimilar, a region that use the originator, but we know that the results in terms of number of sites, etc., are the same. This is a very interesting paper of Oviedo that want to help us to reflect on this. And it's very important that what says Oviedo. They say that we have to evaluate very well different group of patients, PCOS patients, poor responder and other, because we have to know that we have good results in ideal patients, but we have to compare the originator with the biosimilar in different group patients. We know that, for example, in Italy, we have older patients. We have a lot of patients more than 38 years old, and we start to try to use biosimilar and originator to know how works all this one. And our conclusion is that the choice by the clinician on the type of SH gonadotrophin used for venous stimulation should be done on the individual needs of the patient subject to the treatment. taking into account the required dosage, the most pleasing and acceptable route of administration for the patient, the treatment duration, the availability of economic research for the national health system and for patients. But I think that the most important word in our medical practice is personalization of therapy in all different specialties. Thank you.
Okay. Thank you, Professor Morgante. Dr. Milicic.
Thank you, Professor Morgante. Very interesting lecture who give us more information about the biosimilar gonadotrophins. Are there any questions of the audience? Let's be short because we are out of time. Please. Ivan, are there any questions from the audience?
Yes, Dr. Milicic, there is one question from the audience. Professor Morgante, can you hear me?
Yes.
Yeah, the question is, how is the use of biosimilar products different than generics and the reference drug? Would you recommend their use over other options, and in what circumstances?
Okay, there is a very important difference between biosimilar and generics. Generics is a term that is not applicable to the biological drugs like FSH, recombinant FSH. We have only biosimilar. And I recommend the personalization of the therapy. In our experience, we have a small increase of the biosimilar, the dose of a biosimilar is the same dose of biosimilar. For example, if you use 150 international units of originator of biosimilar, we know that biosimilar works more than the originator, and this can be an help or a bad thing, because if we have a patient that could be a risk of ovarian hyperstimulation, it's better to use a drug less potency. But we have a poor patient, probably. We have no studies for this. We have a protocol for poor responder, and we can compare the differences between, in terms of results, between originator and the biosimilar. The personalization is the word. It's very difficult to tell you use the biosimilar or use the originator. Every patient has a different story and a different good protocol.
Thank you, Professor Morgante. There is no other written question. If somebody from the audience wants to ask a question, could raise a hand or could write it down on the chat. Dr. Milicic, there is no other question from the audience.
Yes, okay. Thank you very much, Professor Morgante. Now I'm closing the session.